The preoptic area can be divided into subregions, including the medial preoptic nucleus, and in all species studied, this is the control center for male sexual behavior. Lesions here can abolish male sexual behavior, and it is sexually dimorphic in size, being larger in males. So while virtually all of the brain is comparable between males and females, there are small nuclei where one can identify the gender of an individual based on the morphology of a brain nucleus. The steroid hormones, which are active here, establishing male behavior, vary. For mammals and birds, it is actually estrogen, which is a breakdown product of testosterone intracellularly, whereas in lizards, it is testosterone. The receptors for these steroid receptors can vary according to gender. The medial preoptic nucleus doesn't only affect males, it affects the onset of puberty in both males and females. The hormones of pregnancy influence neurons here, which help the onset of maternal behavior after birth. This is known from studies in rodents. It synthesizes GnRH, which then stimulates the pituitary to make both FSH and LH, essential in both males and females. It is the site of oxytocin production and produces oxytocin receptors, although this seems to be more of a locally acting signal. And it is also the site of AVP production, and there are gender specific levels of AVP receptors. 20 to 30% of the neurons of the medial preoptic nucleus respond to temperature. They are sensitive to warmer temperatures and thus serve as the major center for temperature regulation in the body. The prostaglandin E2 is a signal used here which promotes fever. And also there are neurons here which regulate the cardiovascular system by activating messages which reach the heart through the vagus nerve. So thus they promote parasympathetic stimulation of cardiovascular tissues and inhibit the sympathetic division.